Hello, and welcome back to biology. Today I'd like to talk a little bit about population genetics as a whole, what it looks like when evolution is being driven by natural selection or sexual selection in terms of the actual genetics. We talked a little bit about traits, different types of traits, how they're inherited, things like that. But remember, nature is full of variation. There's all kinds of different things going on. And actually, this is a really cool picture because we had talked a little bit about various traits and things like that in terms of being polygenic, that polygenic inheritance and continuous variation. This is a milk snake, and you can see that it's got the striping pattern, the black, and with if you have a mutation in the snake for not have that, it looks like this. This one doesn't have black or orange. This one doesn't have black, right? And so I think that's kind of interesting in terms of genes. But anyway, moving on. So if you have natural selection, it seems to be reasonable to think in your mind that there might not be any variation if you have natural selection pushing to, have these certain traits more successful, right? And so eventually what you think you should see is a reduction in variance, right? And so stabilizing selection. But this is not what happens. And the question becomes why? And it turns out that two guys, Hardy and Weinberg, not together, solved this problem, right? And so what happened was both Hardy and Weinberg, so now it's called the Hardy-Weinberg equilibrium because of that, showed that the frequency of alleles in a population should stay the same, right, if there are no pressures acting on it. And so the idea that you may have different genotypes, right, so because remember you have two alleles, that you have different genotypes, that's just the combination of alleles, and that can change from one generation to the next. But the actual frequency of the alleles, let's say you have 50% dominant and 50% recessive, that will always be true regardless of how many homozygous dominant, homozygous recessive, or heterozygotes you have, right? And they also showed that dominant alleles don't actually replace recessive alleles, even though you have these changing genotypes. And we'll look at this a little bit more. Okay, so your first question is this: which of the following is not a condition for the population to be in Hardy-Weinberg equilibrium, meaning that the allele frequencies are not changing over time? A small population, random mating, no mutations, no migration, or no natural selection. Well, random mating, that means that you're not having sexual selection. Mutations, no changes in the alleles. Migration, no new alleles coming in or leaving. And no natural selection, no selective pressure there. You do, however, need to have a large population, and Hardy and Weinberg actually called it, and one of them called it a, quote, infinite population, end quote. Obviously, you don't have an infinite population of anything, but the point is that you can't have a small population. Okay, so when we start making predictions, Hardy-Weinberg is actually really useful to try and figure out what's happening with a population. And so we look at the alleles. We're going to call our allele A, right? The dominant allele is capital A. The lowercase allele is lowercase a. We use P and Q in these. So P is the dominant allele, right? Q is the recessive allele, and you can see that I have color-coded them. Yeah, yay. So dominant, recessive. The allele frequencies together should add up to 1, or 100% of the population. So that means if A has a frequency of 46%, then the allele in population B has to be 54%, and that maintains 100% in the population. If allele A changes to 40%, you can expect 60% of allele little a, the recessive. So what does this mean? When you start looking at it in terms of genotypes as opposed to just alleles, which is actually more helpful sometimes, sometimes not, but there you go. You'll have to do it, look at this in terms of each of the various genotypes, okay? And a Punnett square is going to be helpful to show where this equation, which is actually a binomial expansion, P squared is the homozygous dominant, 2PQ are the heterozygotes, and Q squared are the homozygous recessive. So when we look at our Punnett square, and this is like we've done before, here you have the dominant allele P, the dominant allele P. Okay, so these are two heterozygotes. Then you have the Q and the Q from the sperm and the egg. So when you start looking at that, remember you had a 1 to 2 to 1 ratio. Well, P times P is what you've got in the homozygous dominant. That is P squared. Q times P, right, P being the dominant, Q being the recessive, is going to be here in the heterozygote and here in the heterozygote. So there are 2 PQs. And then little a is Q, Q, or Q squared. And so the frequency of the homozygous dominant is P squared. The frequency of the heterozygote is 2PQ, and the frequency of the homozygous recessive is Q squared, right? And you can just see that very standard distribution of alleles among the egg and sperm is what's giving us this equation, right? And so this should be all of the population. Okay, so let's do a sample problem. Let's say that we have a population of 100 jaguars. 84 are spotted and 16 are black. Okay? Based on the phenotypic frequencies, can we figure out what their genotypes are? Well, we have 2 different phenotypes, so that should tell us, is this complete or incomplete dominance? This is complete dominance because that means that the homozygous dominant and the heterozygous both have the same phenotype. Okay? Now, here's something you probably want to know. Black is recessive, right? Now, because heterozygotes and homozygous dominant look the same, we don't know how many of these 100 jaguars are homozygous, or sorry, 84, wow. 84 jaguars are homozygous dominant, or how many are heterozygous. But we do know that all of the black jaguars are homozygous recessive. So we're going to start with that. So our total population is 100. Our spotted population is 84. Our black population is 16. They are all little s. So we'll say spots, because black is recessive. So what that really means is they're all QQ, right? Well, Q squared then equals 16 over 100, or 0.16. Now, we're trying to get all of the genotype frequencies, right? And so we've got to go from the homozygous recessive. We want to figure out the heterozygous and the homozygous dominant. But to do that, we're going to need P, which we don't have now. So this is the goal. We take our Q squared, we get Q squared, but now we need to get Q, because remember, P plus Q equals 1. So we're going to take the square root of Q to get, or Q squared, to get Q, which turns out to be 0.4, because it's the square root of 0.16. Okay, now we've got Q, so now we can rearrange that equation, P plus Q equals 1, to be P equals 1 minus Q, which would be 1 minus 0.4, which equals 0.6. So P is equal to 0.6. Then we can take P and figure out the homozygous dominant, P squared, right? And P squared is 0.6 squared equals 0.36. And that is the homozygous dominant. Now we can do 2 times P times Q, which this should be yellow, by the way, is 0.6 times 0.4 equals 0.48. And that is the heterozygous. So let's check our work. So we had P squared homozygous dominant, the 2 heterozygotes from the Punnett square, and then the Q squared. Now this will give you the total in the population. You don't have to worry about the 2. That's what the equation is now. Okay. So 0.36 plus 0.48 plus 0.16 equals 1. And that's what it should add up to. Now sometimes because of rounding, this may be a little bit less than 1 or a little bit more than 1. There's a little leeway there for you, but it should add up to 100%. Yay, we did a great job. Okay, sample 2. You have black squirrels and you have regular old little gray squirrels over here. So in 1986, a man named Henry Horn counted 133 gray squirrels and 25 black squirrels at Princeton University. In 1994, there were 43 gray squirrels and 9 black squirrels. First of all, you want to think to yourself, what happened to the squirrels, right? Probably something, they lack of food, maybe it was a harsh winter, maybe there were more predators, maybe we cut down trees, who knows? But that's an interesting question, why are there so few? Another interesting question is this population in Hardy-Weinberg equilibrium? Now the first thing that we need to know is which one is dominant. And it turns out that the black here is dominant. It is a less helpful trait, and so it is going to be less beneficial right, in hiding, camouflage, because it's so dark. It's obviously better than albino, right, but it's not as good as gray. So the gray squirrels, even though that's the recessive, ends up being was I going with that? Oh yeah, ends up being the homozygous recessive. So let's look at that. Your total population is 158, right? 133 are gray, 25 are black. But remember, gray is homozygous recessive, so we're going to use that. Q squared is 133 over 158, which is 0.841. We're going to take the square root so we can get Q, because we've got to get P. So that turns out to be 0.917. So that's the homozygous recessive. This one is Q, right? The recessive allele. Okay. Now, right? Now we just do it again for 94. We've got 52, 43, and 9. So 43 over 52 is 0.827, and the square root of that is 0.909, right? So there's our homozygous recessive. This is our Q. And so are they in Hardy-Weinberg equilibrium? No, right? Why not? Well, the allele frequency changed. It decreased. It went from 0.92 to 0.90. Now, that may be statistically insignificant. It depends on what you're doing, but in this case, it's not. So the recessive allele has actually decreased. Okay, why? That's a really good question. You know, maybe squirrels are being protected more. Maybe the campus is sootier or something like that, so those squirrels blend in better. Who knows? Well, I mean, obviously Henry Horn probably knows, but that's not important. So we need to think about why. But basically, there is a pressure that's on them. So if you were to make a prediction, you could see this decrease and start to say, well, if this is the direction in which it's going, then the recessive allele is going to continue to decrease until you reach some sort of equilibrium state. Okay? So Hardy-Weinberg says that allele frequency will not change. That should say frequencies, my bad. But it does. Okay? Which of the following is not true? Allele frequencies often change in natural populations, with some alleles increasing in frequency and others decreasing. The Hardy-Weinberg principle gives a baseline to measure change, and by examining how various factors change the proportions of homozygotes and heterozygotes, we can identify the pressures affecting a given situation. I think I read that poorly. Anyway, you can read. Or all of these are true. Well, the allele frequencies do change, right, in natural populations. The Hardy-Weinberg is important because it can tell you if something is changing, because it gives you a baseline. It gives you like normal, if you will. And then if you examine the homozygotes and heterozygotes and how they're changing as various factors change, you can actually identify what the pressures are in any given situation. With the squirrels, obviously, the gray was no longer as fit as the black, and so you could brainstorm reasons for that. So all of these are true. Now, let's link that to the selection that we were talking about when we talked about natural selection. So directional selection, remember, that's pushing towards one end. What that means is that one homozygote has a greater fitness, and eventually you will lose variation pushing towards one of those homozygotes exclusively, right? And that's called fixation, as you end up getting, so like all of the alleles would be dominant at some point, or recessive depending on which way you were pushing. In stabilizing selection, right, the heterozygote actually has greater fitness, and this is called overdominance. And this is really interesting because it actually reinforces variation in the population, whereas directional selection really decreases and loses variation, that fixation idea. And then disruptive or diversifying is where the heterozygote is the lowest fitness, that's underdominance, and this maintains variation, but it really tends to be very unstable, and those populations will have a tendency to separate into the two extremes. Either the homozygote, like the homozygote recessive population, will move off, and the homozygous dominant will move off over time. It doesn't have to, but usually that's what happens. But this is how those selection types relate to the genotypes. Okay, and then Hardy-Weinberg does not apply anytime you have mutation, gene flow between populations, genetic drift in a very small population, if you have non-random mating, or in the presence of natural selection. Okay. All right, that is it for these notes. If you have any questions, and I have to imagine that you probably will, please ask me in class, and I'll see you next time. Thanks for listening.